Good evening, everyone, and thank you so much for joining us at a fireside, literally fireside chat with Moderna founder, Dr. Derek Rossi. I am absolutely thrilled to have Derek with us this evening to talk about everything that we need to know about mRNA, this incredible vaccine, what the rollout means for our return to normal, and what other really important therapeutic applications mRNA technology is going to lead to in the future beyond COVID. And we are looking at a world beyond COVID. And so I want to highlight also a discussion that we're going to be hosting in early January, January 7th, with Johns Hopkins University, with a great group of panelists. And we're going to focus on the vaccine distribution and access ethics, critically important. We wanted to get this vaccine. There was a lot of concern about when is the vaccine going to come, and now making sure that that it gets to people in an ethical and appropriate manner. We're not going to cover that this evening, but please mark your calendars for January 7th. But before we get into the discussion, it's my great pleasure to share a little bit about Dr. Derek Rossi. He has an extensive and very impressive resume. I first met Derek in 2010. We had just partnered with the Robertson Foundation to award NYSCF Robertson Investigator Awards to the top early career stem cell scientists around the world. We asked the applicants to include their 5 most significant publications. And at the top of the list was a paper that was about to be published in Cell Stem Cell, in which Derek showed that it was possible to use mRNA to reprogram stem cells to be pluripotent, or to turn into different kinds of cells. This was a major discovery for the stem cell field, and we were delighted to welcome Derek actually into the very first class of NYSCF Robertson Stem Cell Investigators. I'm also delighted to share that he just joined the NYSCF Research Institute Board of Directors. Currently, Derek is President and CEO of Convelo Therapeutics, a company that he started with another impressive NYSCF Robertson investigator alum, Dr. Paul Tazar at Case Western University. But Derek's work with mRNA had far broader implications, leading to the founding of Moderna, of course, in 2010, which is a company that really was dedicated to leveraging the mRNA technology as medicine for a variety of diseases. I don't think Derek was thinking of a pandemic vaccine when he founded Moderna. But today, Moderna is a household name, and they've developed an extraordinary vaccine for COVID-19 in record time, and that is thanks to the mRNA technology. And we were delighted to hear just today that the FDA has reviewed their phase 3 clinical trial data and found the vaccine to be, I believe it's 94% effective. And that means that approval is imminent. So we're in a very fortunate situation where so many COVID-19 vaccines are advancing quickly through clinical trials. Here is just a very quick snapshot of the amazing progress, and I'm sure in another day or two that chart will change. Moderna is one of 2 companies that are leading the vaccine race. Of note, the only vaccine to be approved so far by the FDA is the Pfizer BioNTech vaccine, which also uses the mRNA technology. Some vaccines by major companies have faltered in the trials, and this makes the success of the mRNA vaccines all the more important. And it also speaks to the potential of this technology for immunization and treatment of many diseases. So Derek is going to join in a moment. He's a biotechnology entrepreneur. He's been involved in multiple successful biotech ventures. And in addition to founding Moderna, he co-founded Intellia Therapeutics, Magenta Therapeutics, Stelexis Therapeutics, and was a professor at the Harvard Medical School and Harvard University, as well as an investigator at Boston Children's Hospital. He also was named one of Time Magazine's 100 most influential people in the world in 2011. So Derek has been busy and, thank goodness, productive for the greater good. And as I said before, Derek is now a decade-long friend of NYSCF, and we've been so incredibly proud and privileged to watch his ascent in both the research and biotechnology and business worlds. Derek, his approach to science, your approach to science is really about pursuing these kind of long shot, high risk, high return ideas that NYSCF prioritizes. And it's a few of those that have led to the incredible successes that we know of today. So it's wonderful to see you. I am delighted to kick off a hopeful and positive conversation on how this cutting edge science that you've helped develop is soon going to curb this devastating pandemic. And as you can see, Derek and I have teamed up to bring the Fireside to you for this Fireside chat. So Derek, welcome.
Oh, thank you, Susan. It's great to be here, and hello to everybody out there in webinar land. Nice of you to join this evening.
So I'd like to start with the very, very basic questions. Can you explain what a vaccine is and how it works? Because there's so much misinformation, and we have been vaccinating people, as we talked earlier, for hundreds of years.
Yeah, it's a great question. It's amazing to me that I've been busy of late talking with reporters almost every day, a few reporters a day from various parts of the planet. And what strikes me pretty early in our discussion, and these are educated, smart people, and they're sometimes science reporters, sometimes not science reporters, but it almost, invariably comes clear that actually they don't understand what a vaccine is. And then if you actually think about it, most of us don't, because we don't think about it. We get our first vaccinations when we're infants. So we don't have the capacity to ask questions then. And then by the time we, reach sentience, and, it's something that, well, I've got it, it seems to work, it doesn't seem to bother me. So unless you're a biologist thinking about this, you don't actually, sort of go back and ask, what is this and why is it working? So I think it's really important to talk about that. And I always try to make clear that, vaccination is one thing, and then the technology that is used to sort of deliver the antigen, and we'll talk about what an antigen is in a moment, is a separate thing, although it's, they're tied together in the vaccination process. So the history of vaccination, as you mentioned, Susan, goes back indeed hundreds of years. To smallpox actually is really the first viral disease that was treated, and really, if you go back to the 15th century China, they're starting to do inoculations essentially with pustules. It's a little bit gross, but the pustules of people that had smallpox, they inoculate, they take a little bit of that, scabrous material, a little bit of the powder of somebody who's had the pox, and put it onto the skin. Often they scratch the skin of the person and they give a little dose. What they're actually doing is giving a very small controlled dose of the virus to the skin of that person. And what they found throughout the centuries around the planet is that usually was sufficient to stimulate an immune response, which would then confer resistance to smallpox when it was encountered, when the person actually got infected with the smallpox. Typically people that had been inoculated like this were protected. So it has a really long history. It takes another turn in the sort of late 1700s, early 1800s, when some farmers actually realized that there's a related disease called cowpox, It's a different, and there's actually a horsepox as well, which is related to cowpox, but they're all in the same family of viruses. And what some people did, and they actually experimented on their own family actually in the late 1700s, was to take a little bit of the cowpox, again, pustule, and scratch it onto the surface of their family members in the hopes that, being a related virus, it might protect against smallpox. And indeed, that turned out to be the case. You would often get a very small, cowpox reaction, but it was sufficient to confer a long-term resistance to the smallpox, which was, traveling around the planet, devastating society and killing a lot of people. So that's, the origin. It was then later discovered that you could, just take the, the virus itself of smallpox or, rubella or polio, and inactivate the virus and, give that again to a healthy person, get their immune system going by sort of seeing at least a non-infectious agent, but nonetheless, the components of the infectious agent. And again, that would stimulate the immune system to confer long-term protection against any number of different infectious diseases. So really, fundamentally, what it is in essence, exposure of a small amount of something that is expressed by the virus. It might be an inactivated whole virus. In the case of the mRNA viruses now, it's actually just a tiny little protein on the surface of SARS-CoV-2. It's actually called the spike protein. I think many people have heard about that now. If you can see my hand, this is kind of what a virus looks like. And these fingers of mine are the spike protein, and it's what it uses when, now my other hand is a cell. This is the protein that meets a receptor on the human cell to initiate infection. So now what these vaccinations, at least the mRNA, and actually pretty much all the vaccines in development right now, they're all targeted at this spike protein because it's so critical for the first stage of infection. And studies with related viruses before it had shown that this spike protein really is the central protein involved in this family of viruses, the first step in their infectious process. So essentially what all the virus vaccinations do right now is they expose the healthy individual to a little piece of the virus, not even the whole virus, just like I said, this spike protein, just the end of my finger here, introduce that into healthy people's cells, have that expressed with the purpose of eliciting the immune system in the healthy individual to recognize that protein as foreign, mount an immune response to it, generate neutralizing antibodies, such that now when that person goes out and encounters the virus for real at the supermarket or the post office or wherever they happen to be exposed to it, now their immune system has already seen it, or seen an important part of it, just the spike protein, remember, and it's ready to mount an efficient response. So there's the whole, essentially what it's doing is it's harnessing our body's own innate immune systems, which is what, by the way, our bodies do every day anyhow. When we encounter, foreign objects, we're constantly, our immune systems are on surveillance for things that are either self or non-self. Self, you don't react against unless, of course, you have an autoimmune disease, which happens to a lot of people. But basically, you do want to recognize and react to non-self, because non-self is often virus or bacteria or something that can cause you some sort of pathology, give you an infection. So our immune systems really, ever since, viruses and cells first met one another, viruses have been trying to figure out new and clever ways to get into cells, and cells have been, cells and bodies have been trying to figure out defense mechanisms. And indeed, we've evolved these, really fantastic and sophisticated immune systems to respond to these essentially foreign agents. So in short, I know that wasn't a very short answer. In short, that's what vaccination, that's the foundation of vaccination.
So when you were describing the early efforts with the pox inoculation, I was just imagining the FDA and thinking how the FDA would be reacting to that when we have all of these incredibly sophisticated trials. When we were talking a couple of weeks ago, I mentioned to you a conversation I had with actually some very sophisticated nurses who were saying, well, I don't know about that vaccine. Would you take that vaccine? And I said, you know, in a New York Minute. And they said, really? Are you sure? And I said, well, it is basically, it's like the hair of the dog. It's the body reacting in a protective way to to keep this bad thing, this invader, out. And one of the nurses said, well, that's kind of like naturopathy. And you know, there's so much of an emphasis now on things that are organic, things that are green, keeping out things that are foreign. And I think it's helpful to understand the immune system and vaccines in a way that that isn't sort of as a foreign invader that we're, you know,
our bodies and our immune systems are doing this on a daily basis. This is what our immune systems do. They recognize self. They don't react to it. They recognize non-self, and they respond to it. So it's interesting to call it naturopathy, but it's really not something alien. Though I would say, and it may have been in the mind of this person that you were talking about, you know, they've never seen an mRNA vaccine before. We haven't, the world has not seen an mRNA vaccine. I will actually say that many of the other vaccines in the pipeline, almost all of the others being developed by AstraZeneca and Johnson & Johnson and this Gamelia, excuse my pronunciation if I got it wrong, Russian Institute and Sinopharma. These are all adenoviral based vaccines, and that's sort of a different kettle of fish. That's actually, so we could talk about that. But let me talk about at least the Pfizer and the Moderna vaccines and what mRNA is, because I think if everybody gets comfortable with mRNA, they start to understand that, okay, this is actually a pretty natural thing that's going on here, back to this sort of theme of naturopathy. So to begin that discussion, I think we have to talk first about DNA. So DNA is a word that is probably recognizable to most people on the planet. Everybody's heard of DNA. And in fact, I think even most people recognize that, you know, it's the hereditary molecule in the sperm and egg. DNA is the thing that passes on heredity. It's the active hereditary molecule. There's no question it is. But it also happens to be in all cells of our body. And it lives in a cellular compartment called the nucleus. It contains the code of life. It's all written, you know, on the DNA molecule, 3 billion sort of units long, these long linear double-stranded molecules. But in its day-to-day life, in every cell of your body and my body and everybody out there, it's a relatively passive molecule. It's not a particularly active molecule. It contains the code. The code of life is on DNA, but it has to be read to do something. It has to give rise to active sort of worker bees, doer molecules that you know, execute on cellular function. DNA is not that molecule. It's the code, it's the book, the instructions are all in there, but you can't actually do anything with it unless you turn it into something. And that something, it encodes many different types of cellular molecules, but the main one is called proteins. So these are the active worker bees of the cell. They're called proteins. They're actually synthesized not in the nucleus, but actually rather the cytoplasm, which is a totally distinct part of the cell. So the question is, how do you get the code that's living in the nucleus and all that information, how do you transmit that information into making these sort of worker molecules, these proteins? Well, the answer is mRNA. So DNA is actually opens up, its code is read by a bunch of protein machinery actually that synthesize a faithful copy of the code onto the mRNA. So mRNA is actually like DNA. They're very related molecules. They're both called nucleic acids. And so the mRNA is synthesized such that it's basically a faithful representation of the genetic code that's contained on the DNA. What the mRNA has the ability to do, it can, if you remember, DNA lives in the nucleus, it can actually migrate out of the nucleus and go to this site of protein synthesis, which is in the cytoplasm, goes to a organelle. It's called an organelle. It's basically a protein production factory, which lives in the cytoplasm. It fits into it at one end, and its code, which it's carrying, which is essentially the exact code that was encoded by the DNA, is read by this machinery in the ribosome to synthesize different proteins. And then they fold up and they go about do the business of the cell. So that's the basis of all of cellular life on planet Earth.
So they're kind of like smartphones that are actually reading code.
Well, its name, I think, is pretty good. You know, mRNA is messenger RNA. It's delivering the code from the DNA, which is in the nucleus, to the site of protein synthesis, but it's the carrier of this code. It's the messenger. It's a and so this is important. So it's a transient molecule. Unlike DNA, which is a very, very stable molecule, that DNA in your cell is there for your whole life. It mutates occasionally, it gets repaired, but it doesn't turn over greatly. It doesn't constantly get synthesized. When mRNA is made and goes to the cytoplasm and sort of fits into the ribosome and makes the protein that it's supposed to, that's encoded by its sequence, it's essentially degraded after that. So it's a very, very transient molecule.
So it's not hanging around doing evil things.
It's not hanging around doing evil things at all. So literally every cell in your body right now has hundreds of thousands of copies of mRNA in it right now. So you are sitting in front of the fire there, your mRNA is getting a little bit warm because Susan, you're sitting a little bit too close to the fire there, but my fire's a little bit farther back. So there are literally, you know, hundreds of millions of copies of mRNA in every cell of our body right now. So that's really important. So mRNA should be as recognizable as DNA in our lexicon as a word, because without it, you know, I say it like this, DNA makes mRNA, makes protein, makes life. I mean, if you're gonna boil it down to the most simplistic form, that's it. So I call it the trifecta of life. It's DNA, mRNA, protein, life. So that's important for that nurse that you spoke to in New York to understand that mRNA is actually in every cell of her body. And she may or may not know that.
Already, it's not being injected
it's not
in some...
Now, the difference, though, is that normally the mRNA comes from the code on the DNA in the nucleus, and it travels out to the cytoplasm to make the protein. Now, in the case of an mRNA vaccine or therapeutic, it's not being introduced to the cell, the cytoplasm, from the nucleus. It's actually coming from outside the cell. So here's my hand, the cell again. The mRNA is coming, you know, it's synthesized in manufacturing facilities, and now it's coming into the cell, it's entering the cell. So the first compartment that it encounters when it gets through the cell membrane is actually the cytoplasm, which is where the protein synthesis machinery is. And essentially every mRNA that's ever going to be envisioned for therapeutics or vaccine, it's going to encode something. It's going to encode a protein. That's what mRNA does. So in the case of these coronavirus vaccines, well, wouldn't you know, what's the protein that it encodes? Go back to my spiky little finger. It's that spike protein again on the end of, and that's all it's encoding. So the sequence was known. It was published by the Chinese in January of what the sequence of the SARS-CoV-2 genome is. it, of course, encodes for the synthesis of that protein, the spike protein. Previous studies had shown that that protein is important for this type of virus to infect cells. So we knew the sequence. If we know the DNA sequence, we can easily make an mRNA out of that. So now we've got a little piece of the spike protein encoded on an mRNA, and that's what's being introduced now from external to the cell into the cell, going to the ribosome, having it made, and then the immune reaction that happens, that why the vaccination is effective is because the protein that it's encoding is a foreign protein to the human body. It's the spike protein. It's not encoded by your genome. That's what then our immune system says, that protein, that does not look like us. That does not look like one of ours. Let us mount an immune response to that. And that's the basis of this.
So do you think that we can expect to see mRNA used for the flu and for other illnesses that that currently have not perfect approaches?
Yeah, in fact, the several companies, including Moderna and BioNTech, and CureVac, they're trying to make yearly influenza vaccines out of mRNA. And actually, to be honest, I think it's going to be a much better vaccine. We've already seen from these clinical studies that have run now by 2 independent companies through phase 3 in this emergency use approval for one, and another one imminent US, and approval in Canada for Pfizer, and in EU and Britain for the Pfizer vaccine, that basically going through these processes safe and effective. What you can do with mRNA or with any nucleic acid, DNA or RNA are very, very easy to synthesize. They can be very, very quickly made. And you can tailor it exactly to the sequence that you're trying to target. In this case, you're exactly encoding the SARS-CoV-2 spike protein. You know the sequence, so the protein that's expressed in the cell by the mRNA vaccine is exactly that protein. So let's just take influenza, for example. So every year, the influenza shot that we get, and hopefully everybody out there is getting their influenza shot. I've had mine this year. You should get it if you haven't. But it's not actually, it's synthesized, it's the process of synthesizing that vaccine. That's not an mRNA vaccine. That's a traditional usually attenuated virus, which means they actually grow the virus up in the lab and then they inactivate it before they give it to people. So it's not, it doesn't replicate, it's non-infectious, but it has all the sort of, now my hand is now influenza. It has all the sort of proteins that would elicit an immune response in you to, give you protection against influenza. The point with influenza is, though, they start making that, the process of making it is so long, they start making it before they actually know what the predominant strain of influenza that particular season is going to be. Now, they're all influenza vaccines.
It's at least 6 months. It's at least 6 months.
Exactly. And they're all related viruses, but they have differences in their, I wish I had painted fingernails or something here. I could say, they got little differences on their outer surface, which are basically differences in proteins. And these are when the immune response is activated, antibodies are made and they bind to that. And that's how you get neutralization. But if you don't actually know the sequence of the virus and you make a vaccine against a related virus, that's why some years we get better flu vaccines than other years, because sometimes we sort of envision that the strain that's coming is closer to what actually comes. And
it's an educated prediction.
So exactly. So in contrast now, if you wait till you know what the predominant strain is, and you can very quickly synthesize an mRNA vaccine against it, you're actually going to have an immune response that's targeted exactly to the strain that happens to be infecting, sweeping across the planet in a given year. So
Derek, between the day that you identify, so in this case with the coronavirus, the day that the sequence was known to actually having the vaccine was how long?
42 days. So that is unprecedented in medicine. So the sequence was known. It was published online by a Japanese consortia of scientists in Shanghai, and the team at Moderna and others around the planet quickly went and found where the spike protein was. They knew what family of viruses this was, so we knew it was a coronavirus. So let's go for the spike protein. Let's find it. Let's get that exact sequence, and then to synthesize a clinical grade RNA, it was ready to go into patients 42 days later.
So here we are. We're in the midst of the holiday season, and everybody is hopefully walking around in masks, not going out too much, social distancing, and life has dramatically changed and brought untold hardship and food insecurity and just terrible economic circumstances to a huge portion of the world. So what do you think we have to look forward to now with one and Moderna hopefully about to come onto the scene and other vaccines behind them? How quickly do you think, assuming a reasonable rollout of these, we will be able to get back to a more normal life, and what do you think that will look like?
Well, it's going to look different depending upon what part of the planet you live in. Unfortunately, this is just the fact of life, and so I'll give you a long rambling answer. Hopefully I'll actually answer the question that you asked me. So in the West, in the US, where we're living, in Canada, in Europe, the rollout is going to happen, and it's a big logistic rollout for sure. But assuming that goes without a hitch, and that includes the manufacturing of hundreds of millions of doses of these vaccines. If it goes off without a hitch, those who want a vaccine should be able to get a vaccine by the second quarter. So, April-ish, May, something like that. I think we're going to see, and rightfully so, the first order of business is to vaccinate those that are most susceptible. So those healthcare workers at the front line, nursing home staff, and elderly people with pre-existing conditions. I would argue that teachers should be the next line of people that get vaccinated. These are the people that are educating our kids. And that's a pretty vital function, police and firefighters and everybody in these types of public service roles where they have to, interact with the public all the time. These people should be getting vaccinated first. And that indeed is the plan, to vaccinate these. So we're seeing it already in December. People are, Pfizer BioNTech has already started to roll out the vaccine. The first person got vaccinated yesterday. It was pretty cool. I was very excited. Moderna's will be close to follow, assuming it gets emergency use authorization approval, which is likely based on their data. But what we're, but for the rest of the planet, and I'm talking about, maybe, Africa and less wealthy nations and continents, quite frankly, that'll be a little bit slower, unfortunately. I mean, basically, we've got a manufacturing roadblock. If you could synthesize, 10 billion or 20 or 30 or 40 billion doses of these vaccines, this year, then you can inoculate the world within the year. That would be fantastic. We just don't have the manufacturing infrastructure, at least for the mRNA vaccines, to do that. I do believe that infrastructure is being built now, and it should be built around the planet. And I'm advocating that whenever I possibly can, that manufacturing is really important, so that the next time, not just the next time the pandemic rolls around, but we'll have the manufacturing capacity to ramp up quickly. That's and by the way, a pandemic will come again. This is not, pathogens are just so good, and viruses are so good. This will happen again.
Well, I think it's kind of rough to think of it as a silver lining, but when you have over 300,000 people who've lost their lives, and not to mention all of the livelihoods, but this is the most abrupt wake-up call that we could possibly have, and to know on the positive side that we have a way to address it, that it's being taken seriously, that the infrastructure is being developed. And while there's a lot of talk now about these early vaccines requiring exceptionally cold conditions and so forth, as it seems as though as the manufacturing becomes more sophisticated, that some of that could be eased, and the handling of them could become simpler, so they could become available to more people and not just to major medical institutions. But
Well,
I want to just
well, Susan, I hope you're right, but the stupidity of humans never, the brilliance and the stupidity never fails to amaze me, particularly those in the pardon me for anybody who happens to be in the political class, but politicians and policymakers have a view on the horizon of several months at best, or their term at best, and what you're talking about is obviously the way to think about it. We know it's coming, we've seen them before, but let's, if we could possibly take it out of the hands of the political class, which I think we can to a certain degree, but yeah, I don't mean to cast aspersion or be negative there, but
well, we've had a very rough go of it.
We forget our history all the time. We repeat ourselves. We let history repeat itself time and time and time again.
There's no question. I think that's one of the things that I think is so interesting about mRNA, and I'd like to know why you chose to work on mRNA, because
Sure.
you weren't thinking about a COVID pandemic.
Yeah.
And indeed,
It's a fun story.
I want to hear a bit about the therapeutic potential. Let me just mention to everyone who has joined us, if you have questions, please submit the questions, and in the chat or the Q&A function, and colleagues are going to be going through them and finding those that, because I'm sure many of you have the same question, that are representative, and we'll get to as many as we can at the end. But so Derek, why
Susan,
there are so many things that
Susan, hold the thought for a second, because I just want to finish one thought on the last topic,
Go
which
ahead.
is really important,
Go ahead.
and that's that though the Pfizer BioNTech and Moderna vaccines are the quickest to arrive, and that doesn't surprise me because they're mRNA and the technology lends itself to that. Nonetheless, all of these other, there are multiple different vaccines in phase 3, which most of those rely on adenovirus. It's a different thing. Let's not talk about that today, but it's just another way of delivering the antigen, essentially. But all of these need to succeed, because back to the issue that we talked about, Moderna has said that they could produce 1 billion doses in this coming year, and Pfizer BioNTech 1 billion as well. It takes 2 billion two doses, so that's 2 billion doses total. But that's 1 billion people that you can vaccinate. But I believe that population of our planet is closer to, what is it, 8.5, 9 billion, something like that. So we need these other vaccines coming. So it's, I'm rooting for everybody, all of these companies with these vaccines in development. I'm rooting for all of them to succeed.
Well, absolutely, and particularly to the point that you made, where when you have very arduous refrigeration requirements and so forth for some of the newer vaccines, having the ones that are made with the the older formulations that still work quite well is really going to be essential, because you will not be able to roll them out either into rural areas or into countries that don't have the infrastructure to to support that. Also, there's a notion that it's not clear how long the protection is going to last, whether we're going to be looking at something that is going to require an annual flu shot type immunization, or if it's going to be more like a one and done, like measles, or or one and a booster much later kind of thing. So, and then also the question's been raised that the immune system is very clever, and might it be a good idea to have different kinds of approaches to this virus?
Yeah, you mean different types of vaccines to elicit different responses. Yeah, that's an interesting idea. So in answer really to the first question is how long is our immunity going to hold for this? The answer is we just don't know yet. So it's a pandemic that's about a year old now. And within that time period, there's been very few cases of people that have actually been infected once and get reinfected. In fact, there's very few cases of that, and they might have, some issue with their immune system. So by and large, though, I would say that because the pandemic is raging so effectively, if you want to say that, across the planet, that people that have been infected have recovered. Many have likely been exposed to the virus, and we have not seen a lot of people getting reinfected. So I think at least over the course of a year, let's say 6 to 10 months, or 6-8 months, 4, 6, 8, 10 months, we could probably say that there's at least immune protection, a durability of the immune response for that. Now, whether or not it lasts 1.5 to 3 to 5 to 10 years, who knows? And you're right. Some vaccines we need to get a booster every so often. Some we need, well, influenza we need to take every year because it's a different strain that hits us. This is why we have to take that. But so this is just something that will be borne out by the data. So we'll know that in the coming, I would say at least by the end of this time next year, we'll know how many people have been reinfected. Now that's reinfected from an actual viral infection in the first place. And to your second point, might the immune system be triggered in a slightly different way from systemic exposure to the whole virus and to the replicated virus itself versus just the spike protein being introduced by an mRNA or nanovirus or whatever it happens to be? I think that's a reasonable thing to speculate upon. Again, we don't know. And our hope is that the immune response is about as good as it could possibly be. Certainly the clinical trials, that and the data that both Pfizer and BioNTech and Moderna has released suggest extraordinary efficacy. So,
well, I remember the smile on Tony Fauci's face was, ear to ear, 90+%. I mean, that wasn't even something that, that was kind of in dreamland, and here it is a reality. Before we move on to the therapeutic aspect of mRNA and kind of what really intrigued you about it, I want to talk a little bit about side effects, because we'll hear about side effects when you get a vaccination. Typically your arm gets sore, and you might get a little fever, and feel kind of off and achy for a little bit. It's a good thing. Okay.
Yeah, you want to feel that. That's right. And indeed, it's been reported for both of these vaccines, both Pfizer, BioNTech, pretty much all vaccines that don't have serious adverse reactions. Fever is very common, particularly at second dose. Both Moderna and Pfizer, BioNTech are 2-dose vaccines. Pfizer, BioNTech 3 weeks apart, Moderna 4 weeks apart. Those numbers or those weeks were just kind of selected randomly. There's no good reason for 3 versus 4 weeks. So headache, fatigue, muscle pain, chills. These are common side effects of even getting your influenza vaccine every year. And really what that is actually a signal, not of a reaction to the delivery agent itself. Now, in the case of both the mRNA vaccine from Pfizer and Moderna, they're both encased in a delivery vehicle called a lipid nanoparticle. So your body could react to the lipid nanoparticle, it could react to the mRNA, and there's probably some local reaction to both of those things.
Can you
But
explain
really,
the lipid nanoparticle?
sorry?
Can you explain what the lipid nanoparticle is?
Sure. It's it's uh, it, it's typically depicted as a sphere of lipid. It's kind of I wish I could draw it. It sort of again kind of looks like this because but maybe without the sort of spike sticking out of it. But it's basically a bubble of um, lipid that the mRNA is encased in. It's when it's formulated, when it's the mRNA is manufactured, and then they have a formulation process that it's you know uh, uh, put inside of these little lipid droplets, essentially, and that's what's used to inject into both the Pfizer and BioNTech and Moderna. They're intramuscular injections, so you get it in the arm like you would your annual um, uh, um, uh, influenza vaccine. But that lipid nanoparticle helps actually deliver it does two things. It protects the mRNA when it's in the extracellular space, because before it goes intracellular, it's obviously extracellular. So it protects the mRNA from enzymes that might degrade the RNA. And there are lots of enzymes in our body that degrade RNA. They're called RNases. But also it facilitates the uptake of the cargo. And the cargo in this case is the mRNA. It facilitates the uptake of that into the cell. So that's what the lipid nanoparticles do. It's a delivery vehicle, essentially.
And so we don't want it to melt prematurely, so it has to be very cold.
Yeah, well, there's a big difference between the formulations of the Pfizer, BioNTech, and the Moderna. So the Pfizer, BioNTech, that's the one that has to be kept very, very cold. It's minus 80 degrees centigrade, and its shelf life outside of that temperature is actually very, very short. It degrades and basically becomes useless. And this is what you've heard about on the news, that we have to be very careful. Actually, the formulation, which it's a similar formulation, but it's a different formulation. When I say formulation, this sort of the composition of this
The
lipid
recipe.
nanoparticle. The Moderna's is much more stable, actually. So it can be stable in a refrigerator for up to 30 days. And it can be transported at minus 20 and stored actually long-term at minus 20. So minus 20 is your freezer, your kitchen, and 4 degrees is your fridge. And pretty much all pharmacies and transport systems that move medical therapeutics around the planet are all that infrastructure for moving things around at minus 20 is in place.
Right.
Moving stuff around at minus 80 is not in place. That's much, much colder, and that infrastructure is not there. So what I think will, I'm sorry, I know I'm getting off topic a little bit, but what I think we'll see is that the Pfizer BioNTech vaccine will largely go to cities that have the infrastructure in place, at these teaching hospitals and large hospitals that have minus 80 freezers. Whereas the Moderna vaccine might be able to travel out a little bit into the countryside because it can travel at minus 20. It's stable for up to a week at in the fridge. So that's a big deal. Now, adenovirus, which is the sort of delivery vector of many of these other vaccines that are coming, AstraZeneca, Johnson & Johnson, and others, these tend to be quite stable as well. Typically, again, though, refrigeration and minus 20 is not uncommon for any type of biologic reagent. This is very standard stuff. So they'll be shipped surely at minus 20 as well, and we'll have, I think I read, up to 2 months stability at plus 4 degrees. So that's good. And again, though, you still need plus 4 degrees. You can't, send them out into the desert at, and I'm talking centigrade here, I'm Canadian, sorry, plus, 40-degree weather is very, very hot,
Right.
100-degree plus Fahrenheit weather. So nothing, biologic reagents don't withstand that very well. So I realize I got off topic about what we were
Well,
talking.
I'm just going to bring us back to something that some folks might be wondering about. when you joined our stem cell community as a NYSCF Robertson investigator, you were doing stem cell research, and stem cell research and mRNA and COVID vaccine. What role do stem cells have to play in this medicine of the future, whether it's the vaccines of the future or the incredible therapeutics that I hope we'll have a moment to talk about?
Yeah, so I mean, I like the science story because I think it's a neat story. I've told it a zillion times. I don't mind telling it again. So we were working we were inspired by a study that was published in 2006 by Shinya Yamanaka. And I know you know Shinya very well and know of his work, but just to let everybody else out there know. So he had made this discovery, this Japanese scientist, that you could convert, a skin cell, a human skin cell, back through developmental time to a very developmentally potent cell type called like an embryonic stem cell. So an embryonic stem cell is a cell that's very early in development when sperm and egg first meet. And some of the first cells that come out of that, those are embryonic stem cells. They have the ability to form any tissue type in the body. So Yamanaka's discovery, I don't want to go too far into it because
no, he made a version of that. He kind of a copy of that didn't require human embryos.
That's right. That's right. So he was able to turn a skin cell into a cell that had full developmental potency without using embryos. Exactly right. So the ethical sort of issues associated with using discarded embryos for medicine and using these cells was basically abolished by Yamanaka. It was a very, very exciting discovery. He goes on to win the Nobel Prize for this discovery.
But how do we get from that to mRNA?
Well, okay. So we were, yeah, I know. So we were inspired by this work, and I had started my lab at Harvard late 2007, and a very early project that we had, and I'm first trained as a molecular biologist, understanding, how, the intracellular machinery works. That's my training. And then I became a stem cell biologist later in life. My postdoctoral training, I studied stem cell biology, started my lab in stem cell biology, mostly, by the way, in the setting of stem cells that give rise to all the cells of your blood. So they're called hematopoietic stem cells, the cells that reside in your bone marrow and are responsible for giving rise to, by the way, your whole immune system and your red cells and all, your platelets and all the cells that we need on a daily basis. So, okay, tangent, sorry about that. So we started working on this Yamanaka project, and we had this very simple idea. We thought, well, let's try to make these cells. So Yamanaka achieved his goal by introducing 4 genes into a cell, and he did so using, believe it or not, a virus. And molecular biologists borrow from the natural world all the time. So he borrowed from viruses. He took out their normal genome. He put in what he wanted to put in, and he introduced these 4 genes, and that's how he got these embryonic stem cells. But that was going to be tough to medically translate because those viruses integrate into the genome and then they're there forever. They integrate into the DNA and then they're there forever. And if we were going to use these cells for patients one day, we really didn't want that sort of alteration of the genome, that permanent alteration of the genome. So we thought, well, why don't we just try to do it a different way? It was a very simple idea, like maybe too simple. We thought, well, and let's go back to what we talked at the top. DNA makes mRNA, makes protein, makes life. So we thought, well, let's just skip the whole DNA part. Let's just try to use mRNA to express these 4 Yamanaka factors, is what they're called, deservedly so. So we synthesized mRNA encoding these factors and introduced it into cells. And the story gets fun and complicated here because what we succeeded in doing is killing essentially all the cells in our dish. And that was not our goal. Our goal was to express these 4 proteins and get these cells to go back developmental time. But we dug up the work of, we realized we had a problem. And oh my goodness, viruses come into this story all the time. The reason that we were killing all those cells was because as we were introducing the mRNA into the cell, I'll go back to my hand as the cell in a dish, and here's the mRNA coming in from the outside again into the cytoplasm. But it was being recognized by defense mechanisms on the cells in the dish that saw this nucleic acid, which typically does not come in from outside the cell. It typically comes from the nucleus. Remember, it's DNA makes RNA in the nucleus. It goes out to cytoplasm. We were trying to introduce it from the outside of the cell, and the cells looked at that mRNA and said, that looks like an mRNA virus to us, or looks like an RNA virus to us. It wasn't. We weren't trying to encode anything viral at all. It was the RNA itself that was being sort of recognized as a virus. Remember, ever since cells and viruses first met one another, cells have been trying to figure out ways to keep viruses out. Viruses have been trying to figure out ways to get in. So it comes to this story. Oh my goodness, we're almost out of time. So I'll try.
I'm going to bring you back.
I'll try to. Yeah, I better bring it back. So anyhow, we did some modifications to the mRNA that had been previously described by others, and we sort of incorporated it into mRNA, and it worked. And we could express any protein we wished. And then it was the basis of, we started to make every protein in our lab using, we called it modified mRNA. Mod RNA was the short form. We made essentially, dozens of proteins. And after we had made many proteins, I thought to myself, well, we can make any protein. And let's not forget, DNA makes RNA, makes protein, makes life. If we could get this into animals and get this into patients, we might be able to use this therapeutically. So that's
your dream, your therapeutic dream for mRNA?
Yeah, well, it was, I was thinking primarily along genetic disease, actually, originally. So
Just very quickly, because we're gonna, I want to get to some of the questions.
Sure, mutated DNA makes bad RNA, makes mutated protein, makes disease pathology, such as cancer or hereditary disease or something like that. So I envisioned that we could come in with a copy of mRNA that wasn't mutated, was actually the copy that you really needed, the patient really needed in their cell, introduce that as an mRNA therapeutic, have the protein that they truly needed expressed, and hopefully impact their genetic disease, which is what I was primarily thinking about. I wasn't thinking about vaccines, and I wasn't thinking about a global pandemic at the time, though the technology is perfect for that. Again, we need to express a protein in the cell. What better way to do it than introduce an mRNA? And now we can introduce it based on these modifications without sort of triggering the innate antiviral response of the cell, and rather get the protein made and then let the body's immune system react to that.
And so from this crazy idea to thinking about genetic diseases and overcoming them, cancer and other diseases, which I am very, very excited to have take the forefront as soon as we've got this pandemic under control. We have some terrific questions. So I've asked my colleague, Dr. Rekha Iyer, to join us. She also has a fire. We're trying to keep
there it is.
Fireside motif going. So Rekha, if you can, I know there are a lot of questions, and we're kind of a minute out of time. If you have time, Derek, why don't we go about 5 minutes over?
Sure, I can do that.
Okay.
Yeah.
So Rekha, the most pressing that we've got.
Yeah, there were about 70, so I'm going to go as quickly as I can here.
70.
So we had a few questions about autoimmunity that I'm going to kind of combine here. So there were questions about whether these vaccines are safe for people with autoimmune diseases, if there's a risk of someone's body overreacting to the spike protein, and whether that might be different between mRNA and traditional vaccines.
Well, I don't think there's going to be a difference between vaccines, introducing the, I can't say actually exactly, but essentially the delivery vehicle, how you express the protein is the first part of the vaccination, then how the body responds is the second part. So I am actually not sure how, people with autoimmune diseases, though, they have immune reaction to specific antigens in their body. They, let's say for type 1 diabetes, there's antigens on the beta cells that, unfortunately the patient develops antibodies against, and it attacks. And that's this issue of being unable to recognize self from non-self, which we were talking about earlier. So that's essentially what autoimmune disease is. When you get that wrong and you start to see self as non-self, and that's bad, it leads to autoimmune disease and pathology. I don't know if there's, I don't know the history of vaccine well enough to know if any vaccines in the past have impinged upon this. I don't believe so, and I have no reason biologically to believe so, because that autoimmune response is already established in autoimmune disease, and I see no reason why this should impinge upon that at all, because it's, the immune systems in autoimmune patients are still functional immune systems. They just have one autoantigen. So one part of it is, one tiny part of their immune system isn't working well. But the rest of their immune system is working very well. Those people are, able to mount an effective immune response against vaccines or against viruses when they see viruses or bacteria, and they're not at all impaired by that. So I really don't have any reason to believe that vaccination should in any way impinge upon, because like I said, we encounter foreign antigens every day on a regular basis, as do autoimmune patients, and it doesn't impact their autoimmune disease. So, but I'm not a clinician. I haven't studied this area well enough. So I would encourage those folks to talk to their clinician about this issue specifically, but that's at least the thoughts that I have from the answer.
Thank you. And to that end, you talked about antibody responses. So what do we know so far about the antibody responses to the mRNA vaccines, like those produced by Moderna and Pfizer? And should we be following those in follow-up studies to make sure, like, if I go get the vaccine, should I get tested for antibodies 2 weeks later? Is that a useful?
Your question, or that question, Rekha, is essentially, how do we know that we've gotten some kind of immunity?
Exactly.
Well, if I may share my screen, I'm going to share my screen, and so let's see if this is shareable. You let me know if you can see this.
Yeah, we got it.
So this is the data that was released by Moderna today, actually by the FDA. And how do we know that it's working? This is how we know it's working. So this data here shows, and I'm waving my finger around, which of course nobody can see what I'm pointing at, but there's these are the patients that were, healthy volunteers that were either given the placebo in blue or the vaccine in red. And on the y-axis, and again, I'm flashing my finger, nobody can see that, but on the y-axis is the incidence of infection. And so what you see and this is over the course, on the x-axis, it's days. So this is about 105 days out from vaccination. What you see is that the people that got placebo, their rate of infection is ever increasing, and if this were carried out for longer. So if you look on the y-axis and you can read those small numbers, it gets to about 2.5% of the cohort that was given the placebo has actually come down with been infected by COVID or SARS-CoV-2 and developed, didn't develop some developed severe disease, but they all were infected. In contrast, if you look at the red line is the people that got the mRNA vaccine. And if you carry that back, at the end point, it looks like to be about 0.1% of that cohort actually was infected with SARS-CoV-2. So that data couldn't be more black and white, more stark. So I would encourage everybody, if they could just sort of grasp what that data looks like. So that's one thing. So that's really, really good efficacy. That's 95%.
That's better than you normally get for a vaccine.
That's extraordinary efficacy. The important thing, the second issue though, is the safety, obviously, and everybody's always asking about this. Well, again, if you look at the safety data and it's in this downloadable PDF, which is about 50 odd pages long. I just got it today. I haven't read the whole thing in detail, but I know basically the highlights. Anyhow, you get the typical vaccination side effects, which we talked about earlier. But of course, or not of course, but all the people that came down with severe COVID disease, all of them were in the placebo group. And I'm waving my arrow here. I don't know if you can see
Yeah,
that.
that works.
They're all in that group. Nobody came down with severe COVID group or in the vaccine group. And unfortunately, the one person that passed from COVID-19 was in the placebo group. So that type of data and that type of protection for such a deadly disease, that should really encourage, that should be so encouraging. And that's a pretty large clinical trial. That's 30,000 patient or healthy volunteer phase 3. Pfizer BioNTech did the same thing. That's now starts to be tens of thousands of people vaccinated with having, the standard kind of side effects of vaccines. Yet the benefit for such a deadly disease and such a deadly virus is so apparent in the data.
Absolutely, it is. And so now that we have these vaccines approaching the market, what do you think are the biggest hurdles to distribution? And, so also imagine this plays out again. I mean, we all hope that this doesn't play out again. We don't get another coronavirus, but what would be the biggest hurdles then to getting a vaccine to everyone who needs it?
During this pandemic?
During this or a next one?
Well, this one's going to be different from those that follow. I mean, the biggest hurdle and the challenge, and let's just talk about mRNA vaccines, but we could talk about adenovirus as well, which are being developed by the other, Johnson & Johnson, AstraZeneca, and others. This is a relatively new technology as well. There's one approved adenovirus vaccine. So this is not like a standard vaccine either. The reason they can do it quickly is because, again, that technology, the adenovirus, allows for a quick, relative, not quite as quick, but close to that speed of the mRNA. But again, it's also a pretty new technology. And like I said, there's one approved, I think there's only one, it's against tuberculosis. So I think in the future, the challenge that we're facing right now is basically first and foremost, a manufacturing challenge. And that's because these are new technologies and the manufacturing infrastructure is being developed. And, Moderna had pre-planned and they, for their other programs, had, developed a manufacturing facility, a 200,000 square foot facility in Norwood, Mass, which is about 20 minutes away from where I am right now. But that's not enough to create enough therapeutic or vaccine in this case to inoculate the planet. And that's what we need in settings like this. So I think we'll see in the coming years a big investment from various governments in the infrastructure for this type of manufacturing, because it won't just be for pandemics and antivirals, but Susan and I talked a little bit about other mRNA therapeutics. We didn't talk about it enough, but other mRNA therapeutics coming online in the future. Similarly, the infrastructure for adenovirus, large-scale adenovirus. So I think in the future, right now the biggest hurdle really is manufacturing, and that's the rate-limiting step.
Well, hopefully we can
manufacture and hearts and minds, right?
Well, absolutely. And hearts and minds is what we're working on tonight.
That's right. Well, I think one last question for the two of you. What makes you both hopeful for 2021?
So much. I'm so grateful to Derek and the other scientists. We have many in the kind of the NYSCF world, NYSCF Robertson alums, NYSCF scientists working in our own laboratories. People have just full steam ahead, are working very collegially on this problem, and it's having results. This is really tremendous, and I feel that we have had and are having a horrendous year, but for those of us who are lucky enough to be able to turn our full attention to work on these problems, it's an incredible blessing to be able to do that. I feel very fortunate to do it, and the results are astonishing. So I think we may still be wearing masks and socially distancing, but we'll be with our friends and loved ones, and we won't be afraid to visit our elderly relatives or our friends who are immunocompromised. Our kids will be able to go to school. And there'll be a realization that life is very precious.
What about
Yeah,
you, Derek? What are you looking forward to in
well,
2020?
you know, thematically the same. Let's face it, 2020 was maybe the weirdest year that any of us living and sentient have ever had the pleasure of experiencing. It's been a hard year, and so although, and I hate to be the, you know, cast a shadow on the hope, but we're gonna, you know, go through some really hard months. Over the next few months are going to be really hard. The devastation of this thing is going to continue, and it's actually going to be really hard, as we could just see the infection rates and the death rates every day are climbing. The winter is a bad time for viruses. Winter is coming, as they would say in Game of Thrones. Though, with that said, the vaccines are also coming. So there's light at the end of the tunnel. It's coming. So what I'm really hopeful about, in addition to, you know, getting back to some sort of normalcy, but, you know, the economic devastation that has been raked by this pandemic, you know, the economic hole on planet Earth is massive. And I don't believe that we've seen the full extent of it, quite frankly. I don't think it's all been revealed. So I'm really hopeful that, you know, those that have been hardest hit by this can just hang on there, hang on as well you can to get past these few more crappy months that we have coming, because they're going to be crappy. But there's, you know, it's coming that we're going to get back to things, and hopefully, you know, mom, pop businesses will be able to get back in business, and people will get their jobs back, and gym owners will be able to go back and have their clientele, and restaurants, the service industry has been devastated by this. So I really, the human suffering, not to mention just in the health and the you know, the suffering of all those whose family members have been devastated by this disease, but the economic devastation is massive. And so I'm really hopeful that we're going to see an end to that this year, thank goodness. So I encourage everybody to hang in there.
Thank you, Reka. I think that's a wonderful message to really take to heart, that, you know, yes, it is, we're all really tired of it. You know, we want to go and just get normal back. But we have to hang on. You know, the next few weeks with, you know, holidays and so forth are difficult. Wear the masks, see people on Zoom, stay home as much as you can. I mean, it's kind of, think about it. If you have the luxury of staying home and doing your work from home, it's pretty incredible, pretty fortunate. But by the time the weather turns, and I think we're approaching the shortest day of the year, but it's only basically one more week, and it's going to start to get lighter. The vaccines are here. They're not coming, they're here. So you're going to go, you're going to get your jab. You might feel crummy for a day, and life is going to move on, and then we can get to do the hard work of repairing all of the damage that's been caused. But I think also we have new leadership that is going to be coming in January as well. That's made working on all of these problems a priority. So I want to thank you so much, Derek, for joining us and for doing the work that you do. Really, it's so important. So who knew that? And I look forward to having you back for another conversation when we can talk about all of the therapeutic things that this incredible discovery of mRNA and the technology that you've helped develop can do. And we will have COVID in the rearview mirror. Not right now, but we will.
Happy to. I look forward to that. But it's been really fun. Thanks so much, Susan, for inviting me, and thanks to all that joined. Do please, you know, if you learned anything today about, you know, how these vaccines work and, you know, why you should go out and get vaccinated, please do communicate that with your friends and family, and encourage everybody to get the vaccine when they have a chance to do it, and stay safe.
Stay safe, stay positive, and we'll be back to talk to you very soon. Thanks so much.
Thank you.